Welcome to Two Bees in a Podcast, brought to you by the Honeybee Research and Extension Laboratory at the University of Florida's Institute of Food and Agricultural Sciences. It is our goal to advance the understanding of honeybees and beekeeping, grow the beekeeping community, and improve the health of honeybees everywhere. In this podcast, you'll hear research updates, beekeeping management practices discussed, and advice on beekeeping from our resident experts, beekeepers, scientists, and other program guests. Join us for today's program, and thank you for listening to Two Bees in a Podcast. Folks, we really have a treat for you today on Two Bees in a Podcast. We have world-renowned professor and scientist Dr. Marla Spivak from the University of Minnesota. She'll be joining us talking about social immunity. That will be followed by a discussion between Amy and myself about swarms. What are they? How do they work? And most importantly, how can we prevent them? Today's episode will conclude with Stump the Chump, our question and answer series. So, Amy, I have been, as excited about this segment as I have been really any of the segments that we've had, because we are going to be interviewing today Dr. Marla Spivak. Dr. Spivak is a Distinguished McKnight University Professor. She is a MacArthur Fellow in the Department of Entomology for the University of Minnesota. If you have been in bees and beekeeping for any amount of time, really, no matter where you are on planet Earth, you will have read some of her writings. You will be aware of Dr. Spivak, and her research will have touched your beekeeping lives in some way. So it's really my pleasure, Dr. Spivak, to welcome you to Two Bees in a Podcast.
Thank you for having me.
Absolutely. It's like I'm almost a kid in the candy store. I'm trying not to be overly
He's fanboy.
excited. Yeah, I know. It's funny because years ago, when my program was young and I was trying to design mine after someone's, Marla, yours was one I was trying to emulate. And I remember asking Gene Robinson years ago, I'm sure he'll never remember this, but I was asking him years ago a question. He's like, hey, well, you should ask Marla. She's got, one of the best programs in the world at what she's doing. And so you're even recognized by your peers similarly, Marla. So thank you again for joining us. I'm excited to have you.
Thanks. It's flattering.
Right, well,
Flattery will get you nowhere, though.
Well, we're, as our listeners know, we're all podcasting from our closets at the moment. Amy is, I am, and so that's the best I can do here from my closet, Marla. All right, so what I want to do is, before we get into the topic of social immunity, which is why we have you on today, I want us just to hear from you a little bit of an overview about your background. How, what did you do? Where'd you go to school? How did you get into bees? Things like that.
I got into bees when I was 18 years old, and I'm not going to tell you how many years ago that was, because it was many years ago.
Just a couple.
Yeah, just a few years ago. And I was not the best student, and I was bored and directionless, I would say, in college. And I wandered into the library to look for something to read, and somehow picked up a book about bees, and I spent the night reading it. I didn't go to sleep that night. Excuse me. I couldn't put the book down. And the next day I told my advisor that I needed to go work for a beekeeper.
No, so let me ask you a quick question. What in the world was that book? Which book was it that made you?
Well, I don't know that it's that good of a book. I have reread it, and it's, it's okay. I don't know what grabbed me at that time. It's called Bees Ways.
Okay.
And the author's last name is Curtis, I believe, or maybe his first name is Curtis. I don't remember. It's not a very well-known book, and he's a naturalist, and it was just beautifully written, actually. Just something about that honeybees are social, and they interact with each other, grabbed me, and that there are these beekeepers that just love their bees, and I hadn't really thought about loving insects that live in colonies that sting you. I just thought it was so weird I had to go see it for myself. So I went to work for a commercial beekeeper, a family that has about 2,000 hives of bees in New Mexico. And I worked for them for 4 or 5 months, I guess. And then I was hooked.
That's pretty incredible. I'm amazed that you just randomly picked up that book. It sounds like serendipity. I mean, that's pretty crazy. So you've had a distinguished career. Have you spent your whole time at the University of Minnesota? How did you end up there specifically? How long have you been there? Tell us a little bit about that.
Sure. I did my PhD at the University of Kansas. Then after
Oh,
that,
hey, I'm from Kansas.
are you?
I am. I went to K-State, though. It's a Wildcat.
Well, KU is
Conversation over.
much better, of course. But after after I got my PhD, I did a postdoc in Tucson at the University of Arizona, the Center for Insect Science, and I was actually based at the USDA Bee Research Lab down there in Tucson. And then I was hired here actually in 1992, and I was hired as a temporary non-tenure track position. But then within a few years, they made it a tenure track position. So I've been here since.
That's really cool. I think they realized what they had, I guess, and worked really hard to keep you. We could talk maybe another day about the fact that you have a new bee lab and all the different programs that you have. But yeah, it's pretty miraculous how you ended up there.
Well,
So
you have a new bee lab too,
Yeah,
Jamie.
we're fortunate. We're fortunate for sure. I'm not
we're
going
gonna
to
have to
lie.
have you. Yeah, we're gonna have you come visit sometime. But to get back on track, let's go ahead and talk about social immunity because that's what we're here to talk about. Can you tell me what that is and why that's important?
Sure. Social immunity is a term that was coined by Sylvia Kramer, who is one of my heroes in research. She actually studies ants. She's from Austria.
Oh, cool.
I believe she's from there, at least she works there. And she coined this term to describe how social insects colonies keep themselves healthy. So honeybees and ants and termites and some of the big social wasps live in very, very dense societies. And so social immunity describes their behaviors or how they organize themselves or their physiologies, what they can do to defend themselves within their nests against parasites and diseases to maintain their health and survival.
Yeah,
So
that's kind of
basically,
amazing.
in a nutshell, it's how they keep themselves healthy, and which is very pertinent right now as we're all worried about getting COVID-19, excuse me, and how we keep ourselves healthy when we live in dense communities.
Mm-hmm. So it's really interesting, just because, I think the normal person would see an ant or a wasp or a termite and not think it, we just want to get rid of them. But once you start digging a little bit deeper into being insects, it's very fascinating. And so do you have examples of honeybee social immunities?
Sure, I have lots of them. One of them that I studied for a very long time is called hygienic behavior. And the fun part is that I studied hygienic behavior, which was a term coined by Walter Rothenbuehler in the 60s. The behavior was described by some beekeepers and researchers in the 30s and 40s. But when Sylvia Kramer coined the term social immunity, all of a sudden, like 10, 15 years of my studies had a context.
Mm-hmm.
And all of my life was like, oh, that's what I do. I study social immunity. How cool is that?
Yeah.
But hygienic behavior in honeybees is the ability of some bees, it's a genetic trait, and it's their ability to detect diseased brood in the nest. So some larvae and pupae that get sick, or maybe that are parasitized by varroa mites. And so some bees in the colony can detect that there's a problem in the nest, and they yank out the sick or parasitized brood and throw it out of the nest. And in that way, they're limiting the transmission of the problem within the nest. So they're basically weeding or culling out the sickness from the nest.
I think it's interesting how you describe kind of your epiphany about this. You now are aware of social immunity. I had a similar moment with it as well. When I was a graduate student in South Africa, I read Robin Morritt's book, Bees as Superorganisms, one that he co-authored, and he talked a lot about this idea of the superorganism concept. So I became kind of fascinated with that, and then read E.O. Wilson and Holdobler's books on the topic for ants, etc. And as I started giving talks on superorganisms, I kind of started discussing, how the investment in the colony is what was important and not necessarily the individual. So what you start seeing are these colony level quote, phenotypes that arise out of these groups of insects. And so when you think specifically about immunity, hygienic behavior is one of those. There's not necessarily a quick and direct benefit to the individual bee from hygienic behavior, but it confers a significant benefit to the colony, right? And when all of that started clicking at me, and I started stepping back, looking at how colonies handle disease or pest pressures or other stressors, I started going, wow, it's pretty miraculous what they are capable of doing. I mean, you have spent a lifetime studying hygienic behavior. You mentioned it, but there's also some other things that you're looking at, even recently, propolis. We'll get into that in a moment. But let's think specifically about hygienic behavior. What do we know? What do we need to know about the benefits of hygienic behavior for colony health?
Sure, but I want to dwell a little bit on the point that you were just making about the superorganism first, and then you'll have to remind me of the question because
yeah, not a problem at all.
But yes, the colony is the animal, if you will, and there's a bunch of little animals within the animal. And so to think about social insects and honeybees in particular, we have to train our minds to jump levels of selection, if you will. So there's selection on the individual bee, there's selection at the colony level, and then at the population level. And I really it's like a mind game, if you will. I really like to jump those levels and think about what's going on in the individual, and inside the individual, its immune system, its microbiome, et cetera, everything that's going on in that individual, and then scale that up to the colony level. Now, the animal level, full of all of the complexities that's within these thousands of little animals within it. And yes, the colony has its own phenotype, its own personality, if you will, and its behaviors and ways that it keeps itself healthy. That's what I really like about really highly social insects, is that kind of thinking.
I agree completely. And I love this concept of emergent properties. When you put two bees together, you've got two bees. When you put 100 honeybees together, you've got 100 honeybees. But when you put 1,000 honeybees together, you've got a colony. Something else happens when you squeeze bees together. Properties begin to emerge that are otherwise absent in the individuals, but the genes and the behaviors of the individuals govern these emergent properties. And I agree with what you said. Love it completely. This idea that we can go back and forth between the individual, the colony, and then the population, and then back down, and consider how certain things have upstream or downstream effects on these three things that we look at: the individual, the colony, and the population. So I agree. So let's, I tell you, we're going to end up getting very philosophical here, I guess, if we're not careful. But let's kind of hone in again back for hygienic behavior. So as a reminder, what do we know about it? What do we need to know about it with regard to colony health?
Hygienic behavior is honeybees, one of their most important mechanisms, I think, to keep the colony healthy. I think there's a number of ways they can do that. But honeybees are really well known for their ability to detect and remove diseased and then mite parasitized brood. Like I said, not all honeybee colonies can do this very well. For me, the most important part of the hygienic behavior is the timing of it. So if we're thinking about diseases right now, if a colony has a little bit of chalk brood, or if they have American foulbrood, there's a disease progression. So the adult bees carry the pathogen, but they don't get sick. So adult bees don't get chalk brood or American foulbrood, but they have the spores of those diseases on them or in them, and they inadvertently end up putting some of those spores in the food of larvae. The larvae then eat that food with those spores, and those spores then germinate inside their guts and then continue to multiply. So those spores germinate into what we call the growth phase or vegetative stage. For a fungus, it would be the mycelial stage. And that grows and grows and grows inside them until it bursts the gut and then kills the poor little developing bee. The interesting thing is that hygienic bees can detect that brood, that larva or pupa, is sick while it's in the non-infectious stage. So while it's in the vegetative stage or the mycelial growth stage, so that when the bee removes the larva from the cell, the adult bee removes the larva, that adult bee is handling something that's not infectious. So then they throw it out of the nest, and then they can not get sick themselves and not transmit the disease. So the timing of the removal is the really fascinating thing. Ants do the same thing. So in Sylvia Kremer's lab, they have postdoc, his last name is Pull, and he studied how ants do this also. It's really fascinating.
Yeah, I'd never heard that explanation. That's pretty interesting.
Yeah, that is really interesting. We'll definitely have to bring you back. I mean, now that I'm listening to you speak, we're going to have to bring you back to talk about all the different research that you've done. But I know that you've also worked a little bit with propolis, right? And so, can you tell us a little bit about that and maybe some of the known benefits of propolis for colony health?
Love to.
Yay!
So propolis or propolis, tomato, I don't know how to pronounce it, is actually
Definitely tomato.
is actually resin. So some plants produce resin, and it's a plant defense. It's a way a plant can keep itself healthy. So these resins are highly antimicrobial. They fight off bacterial and fungal and even virus pathogens in the plant. Bees can cue in to these plants that are producing these resins. And in our area, it's mostly cottonwood trees on the leaf buds. And they go into the cottonwood leaf bud and scrape the resin right off the bud and pack it in their hind legs and then bring it home to the nest where they use it like a cement. And they'll put it in cracks and crevices where beekeepers know and love it very much, I'm sure, because it gets in makes it very hard to pry boxes apart and pull frames out of the boxes. It's on all of my clothes. Propolis is everywhere. But it's in the same way that it's a defense or medicine, if you will, for the plant. It has the same function inside of a bee colony. So resins equal propolis, but propolis in the nest sometimes is mixed with a little bit of beeswax. But basically the chemistry of the resins are intact, the same as they would get it from the plant. And we know that it has many, many functions to help bee health. And it's really important to the bees. As beekeepers, we've been selecting against colonies that collect a lot of propolis because it gets in our way. But I think we've missed the boat on that one. I think that it's important for the bees to have propolis within the nest to support their immune system, to support their microbiomes, to help them fight off pathogenic bacteria and fungi that are in the nest.
One of the ways that I was introduced to propolis use the most is, even though I've been keeping bees for a very long time, when I did my PhD in South Africa, it was pretty overwhelming to me, the propolis use of the bees that I was working with there. They would take standard Langstroth hives and the bees would reduce their own entrances to about an inch. We did some subsequent work there where we were working with wild honeybee colonies, some nesting in cliffs. They would close, if you think about it, if you're nesting in a hole in a cliff, you've got this exposed hole. Well, they would close off that entire hole, except for with maybe a centimeter or two, with an entire sheet or a wall made of propolis. I mean, so there are lots of honeybees out there that still use copious amounts of propolis. And I don't mean just a little, I mean a lot of propolis, reducing entrances, building walls with it. It's pretty amazing what wild honeybee populations do with this stuff.
Do they eat the propolis?
No, it's well, Marla, I'll let you answer.
No, it's fine. It's fine. No, they don't eat it. And the smart bees that you're talking about, resin would be hard to get down their little esophagus, I would think.
Yeah, sure.
Yeah.
Gum up your mouth parts. But yes, propolis collection is a heritable or genetic trait, and there's some races and kinds of bees that really, really bring in a lot of propolis. Bees from Africa are really notorious for bringing in a lot of propolis. Stingless bee colonies from the tropics also use a lot of resin. That's part of their nest structure, is they encase or enclose their combs with resins. So it's important.
So when I consider social immunity, you gave us two examples today, hygienic behavior and propolis use, and then I try to get all this back to a beekeeper. Are there some strategies that beekeepers can use to maximize the social immune response of honeybees? And when I give lectures about beekeeping, I always start off saying the best beekeepers are those who know what the bees are trying to do. They know what they are trying to do, and they work to harmonize the two. And you just said it, we've been breeding bees for reduced propolis use. Well, bees are trying to use propolis perhaps for colony health purposes. So what are strategies beekeepers can use to maximize the social immune response of honeybee colonies rather than work against it?
Yes, it's very important to follow the bees' lead instead of trying to impose our will and our lead on them. Mm-hmm. They don't read the books, so they're going to do what
they don't.
They're going to do what they want to do anyway. But I think it's important to then, for beekeepers, as far as propolis is concerned, maybe consider using boxes that have a rough interior, unfinished lumber, or even gaps and very rough textured inner surface of our bee boxes. And that will encourage bees to put propolis where they like to do it naturally, as they would within a tree cavity where they would nest naturally. I think that's one way to help them do what they would like to do, and maybe not put so much propolis where we don't like it. As far as hygienic behavior, if that's pretty easy trait to select for. The assay for it is pretty easy. You freeze kill some brood, and then check up on it within 24 hours how much of the freeze killed or dead brood the bees have removed in 24 hours helps you get toward bees that are hygienic, that will detect and remove diseased and mite parasitized brood. If you're really looking for mite resistance and hygienic behavior, there's better assays, and that's a whole discussion on Varroa sensitive hygiene and how you select for that. And if we go there right now, we're going to get deep in the weeds.
We'll get in that for the next podcast that we have with you. So I guess, what are some things that beekeepers might do that may harm or limit a colony's response? I know you kind of touched a little bit on that just now.
Well, for propolis, I think it's trying to scrape it all off and get rid of it.
That's the first thing we always do, right? We just scrape the propolis right off whenever we open that box.
Well, and what I say to beekeepers, if it's in your way, yes, scrape it off. I mean, if it's impeding your management, then get rid of it. But you might consider that the bees are bringing it in for a reason. It's super hard for bees to collect resin. When they get it on their back legs, they can't even get it off their own legs. Another bee in the nest has to remove it, and they yank it with the removing bee grabs it with her mouth parts, her mandibles, and pulls, and it's sticky. And you can just imagine trying to deal with this stuff within the nest. And yet they continue to bring it in. So that was what made me realize, the bees really want and need this stuff. Mm-hmm. So maybe we should be letting them bring it in and put it where they prefer to put it.
Well, Marla, let me tell you some of the things that I've been thinking about recently. I was asked about a year and a half ago to provide a chapter in a book that's being written by some Italian colleagues of mine. It's called Welfare of Managed Honeybees. And the premise is this. The OIE in Europe has defined welfare with 5 points. And they make the argument that essentially animals that are under our management have certain rights. For example, think cattle or poultry or what have you. And they have the right to be, free from disease, the right to be free from fear or malnourishment. They have the right, basically, if we're going to engage in husbandry of these things, there are certain standards that we should follow to make sure that there are not undue stressors added to those animals' lives. And so he was wanting us to take that same perspective for the honeybees, right? We've got these colonies, but we do things to them that are stressing them. And we also limit their ability to address stressors the way that they should. So as I think about the welfare of managed colonies, one question that comes up are what obligations do we have when we keep bees? Like if we are going to take a bee colony and put it into our yard, do we have certain obligations to ensure its health, to take care of it and make sure that it's free from stress? In other words, make sure that its welfare is taken care of. And when I listen to you talk about social immune responses and hygienic behavior and propolis use, I think that, in many ways, we probably have an ethical obligation to let the bees do what they need to do to address these issues that they have, including collect and utilize propolis. I mean, what do you think about that? Am I going a little too far or maybe not far enough?
Yes, it's a very delicate balance, isn't it? Because honeybees are not really domesticated like livestock, and yet they're really not a wild animal if you put them in a box and manage them. So where do they fall in that spectrum? It's really interesting to think about. But I think if you are a beekeeper and you keep bees in a box, then you have some responsibility for their health. You have the responsibility to make sure that they don't spread diseases to other colonies in their community and that within the 2-mile flight range. That's their safe or social distance, if you will, really. And yet you also have the responsibility to allow them to take care of themselves when they can. So our bees in the United States aren't fully able to take care of themselves against the varroa mites and associated viruses that the varroa mites might be vectoring. And so in that case, it's our definite responsibility to keep the mite levels, control them in some way so that they don't get out of hand, which then not only kills your own colony, but puts other colonies within flight range of yours at risk. So it's more of a community level thinking that we as beekeepers need to be doing. Yes, that's a very interesting and complicated question of how beekeepers find this balance between managing their own colony and making sure all the neighbors' colonies are within flight range are not put at risk, and yet allowing the bees to do what they need to do to keep themselves healthy.
Yeah, it was a very new concept for me, this idea of welfare of managed honeybee colonies. And the reason I bring it up in our discussion is because the chapter that I wrote for that book required me to read a lot of your work and others about social immunity. And so it was kind of interesting because it seemed to go hand in hand. What bees are trying to do naturally to address their issues, we need to permit it. But what bees are incapable of addressing currently, we need to help. I feel like we have an ethical responsibility to take care of the bees the best that we can, which of course, as you know, will bring up interesting discussions between certain groups of beekeepers down the road. Nevertheless, Marla, you've been a fantastic interviewee today. Thank you so much for joining us. I appreciate the time that you put in here as we're all kind of hunkering down in the midst of COVID-19. Just thank you for taking the time to be with us this day on Two Bees in a Podcast.
That was fun. Great questions. Thank you so much.
Good. So audience, you've been listening to Dr. Marla Spivak, who is a Distinguished McKnight University Professor and MacArthur Fellow in the Department of Entomology for the University of Minnesota. Thank you so much for joining us on Two Bees in a Podcast.
For more information about this podcast, check out our website, ufhoneybee.com. All right, in this section, we are going to talk about swarming. Jamie.
Yes.
Are you there?
I'm here.
It's so weird to podcast without you sitting next to me.
I know it is a little odd, but I promise I'm here listening.
That's fair. So it's springtime, and this is really a time where a lot of beekeepers start seeing swarms, catching swarms, finding swarms. People who don't like bees start to see swarms, and they really want to get rid of them. So we figured we'd just do a little segment on swarms and what it is and why bees do it. So can you just go ahead and tell me, what a swarm actually is?
It would be my pleasure. So, honeybee colonies reproduce by swarming. That's the basic principle of swarms. So let's pause for just a second. A lot of beekeepers, and myself included when I was young, think that colonies reproduce in numbers of bees. And if that were the case, then colonies would just continue to grow and grow and grow and grow and grow until they get infinitely large, as it were. But really what a colony wants to do is not reproduce in numbers of bees, it wants to reproduce in numbers of colonies. And so, Amy, the way that I almost always teach this is things that can reproduce want to really, really bad. Reproduction is one of the strongest drives in all organisms. So if swarming is colony level reproduction, then it is an incredibly strong drive of the honeybee colony. And that is, in fact, the case. In fact, everything that a bee colony does is to prepare it to swarm and enhance the chances that that swarm will survive. And so think about it this way. When a colony is coming out of winter, it goes into growth mode, right? There's a lot of incoming nectar, there's a lot of incoming pollen. That colony begins to grow and grow and grow and grow. Somewhere in that process, they'll start producing drones. All right. So also in that, and they produce drones. If you think about it, they're producing drones because it's reproduction season. There's going to be a flush of virgin queens from their colony and other colonies as well. And they want to make sure drones are available. Then they grow to fill the capacity of the nest in which they live. So they're starting to get congested. The daylight is getting longer. The temperatures are getting warmer. The food sources are available. So then they will start building queen cells. And at that point, they will put the queen on a diet. They'll actually get on her back and shake her and cause her to run. So we just...
What? Really?
Yeah, they do. And Amy, guess what? Diet and exercise causes the queen to lose weight. And that's necessary. That's necessary because it's the old queen who goes out with the swarm. So they have to get her down to her flying weight, which she's not at while she's laying eggs.
Okay, wait. I have a question though.
Oh, shoot.
If you have a queen excluder and she starts to slim down, do they get her small enough to go past that queen excluder?
In some cases, Amy, they actually can. See, a lot of people will try to stop swarming by putting excluder material on the front of their colonies, but that's only moderately successful because a lot of times when queens are slimmed down, they can fit through those excluders. Not all the time, but a lot of times.
Sure.
So what happens is when they get her down to her flying weight and they, quote, see that there's queens being produced, about 30 to 70% of the workers will rush out of that hive. The queen goes with them. They'll all fly around in the air. They will all land on some structure, a tree limb, a fence post, etc., and they will sit in that structure looking for a home to which they can move, right? So basically, when you're watching a colony swarm, you're watching a colony give birth. That cluster that's hanging on that tree limb, it's a baby bee colony that has to send out scout bees to go and find new nest sites, and then it'll fly to those new nest sites, inhabit it, and so on. What's left behind at the parent colony is you've got this hive that's got a lot of queen cells. The first queen who comes out, she'll go around and bite holes in the queen cells of the other developing queens. She'll stick her abdomen in there and sting her competition to death. She then becomes the reigning matriarch in that hive. So essentially, that colony then came out of winter turbocharged to try to swarm. And then you've got two colonies now. You've got this one that's hanging in the tree that's got to go find a new nest and has to store honey to survive the coming winter. And you've got the parent hive that now has to rebound from losing all those bees and its queen, store honey, and survive winter. So they do their activities the rest of the year are to prep themselves to survive winter, which they do to try to swarm. So you could argue that everything that a bee colony does is to make it possible for them to swarm, i.e., possible for them to reproduce themselves.
So you're talking about them choosing, like the bees looking for a new place to swarm and to live. Do they make that decision? I'm sure you've read the book Honeybee Democracy.
Yep, I have.
But it talks a little bit about, their decision making and where they're going to go and how they kind of influence everyone as a whole. So are they doing that in the original hive before they go and swarm, and then they do it again when they look for their final destination?
So that's a great question, Amy. It's spot on. And it's funny. It's almost serendipity because about a week ago, maybe 2 weeks ago, someone emailed me and asked me, did bees look for new nest sites prior to leaving the nest in which they live? We know they do it from the swarm cluster.
Sure.
We know that is the case. The question is does it start earlier while they're in the hive? And ultimately, that question was passed to Tom Seeley, and I was on the email chain. And Tom Seeley, of course, is the professor at Cornell University who wrote Honeybee Democracy and was, without
Oh,
question, the
cool.
world expert on swarming behavior. And his reply is that there is evidence that bees do begin scouting for nest sites prior to leaving their nest. And then once they swarm and form that cluster hanging from a tree limb, as it were, they continue that process, amplify it, make their decision, and move from there. It is my desire, Amy, to have actually Tom on as a guest in our podcast. Well, Tom, if you listen to our podcast, we're coming for you, buddy. But
I'll have to send this over to him.
but I will say that that his research has been, paramount to understanding what goes on in swarming behavior. But
Sure.
so, long story to answer your question, yes, there is some emerging evidence that they do in fact start that process prior to even swarming.
That's really amazing. It's like they're out, looking for a new place while at the same time they're swarming. And so they're just having to catch up and keep up with the rest of the colony at that point, right?
I think, Amy, one of the cool things about it is there is so much to this story, how they prep for it, how they
Sure.
move, how they find the site, and once they find the site, how they move in unison. And we really need to have Tom on to just go into the great depth of, how does all of this process work? Because it is mind-boggling and will blow the socks off beekeepers listening to this. It's incredible what bees do during the swarm process.
Yeah, we'll definitely have to have him on sometime. But so just real quickly, how long do swarms typically stay, in their first stop, I guess? Because a lot of the calls that we get are, we found a swarm and, maybe 20 minutes later, 30 minutes later, they're gone. So is there a general time period?
That's important. And the reason is important for the very question that you just asked, right? We get a lot of people saying, oh gosh, we've got this swarm that just landed on our property. What should we do? Should we contact a pest control operator or a beekeeper or what have you? And in reality, that holding cluster usually only lasts a couple hours in the best conditions. When it's sunny and there's a lot of available cavities and the weather's good and it's not raining, the bees can do their job. They can look for these cavities. They can scout them out. They can move to it. And this can take as just a few hours. On the other hand, if the weather's bad or the cavities aren't so available out there in the wild, or perhaps it's raining or too cool, what have you, those clusters can sit there all day, sometimes 2 days or 3 days. I have seen it happen so long, in fact, that the bees almost just give up and they just start constructing their new hive where they are hanging. Obviously, that's not ideal because the colony then would be exposed to the elements because they don't build a shield around themselves. But generally speaking, it can take a few hours to a few days in the best of circumstances.
I'm just imagining like the new colony kicking the old colony out and saying, we don't care where you go, but you can't stay here.
Yeah, there's a song that comes to mind, but I shan't sing it. Number one, because I don't sound good. Number two, I don't sound good. So maybe
that's fair. Okay, so for my last question before we wrap up, why is swarm management important, for a beekeeper?
Okay. It all has to do with swarm biology and the timing of swarms. Generally speaking, swarms happen a few weeks before or during the first half of the major nectar flow. Now, think about it from a biological perspective. This colony, this swarm, is leaving its parental nest where all the resources are. This swarm is not carrying with it wax. It's not carrying with it lots of honey. It's not carrying brood. It's not carrying lots of bee bread. Essentially, they have nothing but themselves, and they have to move into a cavity, and they have to provision that cavity by building wax. They have to store, somewhere between 50 and 100 pounds of honey, and they have to do all of this with the remaining nectar flow. And if the average major nectar flow lasts 4 to 6 weeks, and they swarm at the beginning of the nectar flow, then they have 4 to 6 weeks to do the bulk of their work to prepare themselves for the winter. That's months and months and months away. So as a result, most colonies swarm immediately before or at the beginning or during the first half of the major nectar flow. What basically this means is they swarm when you as a beekeeper least need them to, right?
Yeah,
you
of course.
need the bees to be there to make honey for you. So they are swarming at the worst possible time. In swarms, not only do you lose a lot of bees very quickly, but you've lost your mated queen. Your your parental colony is the one that has to make the new queen.
Mm-hmm.
And just to do some simple math, I'll round everything up to a week to make the math and the discussion easier. But if they leave,
It's probably because I'm really bad at math.
well, that's because I'm bad at math. I need to make it easier for me to think. If they leave with capped queen cells, that it'll take about a week for the first cell to emerge. It'll take about 2 weeks for that queen to mate and lay her first egg. It'll take about 3 weeks for that first egg to emerge as a worker. So basically, you lose somewhere in the neighborhood of 6 to 7 weeks of brood production when you have to requeen your colony that time of year. And if the average major nectar flow lasts 6 weeks and the average rebound from a swarm is 6 to 8 weeks, you are going to lose a lot of your honey flow. So bees are most wanting to swarm when you least need them to, which is why those beekeepers who are so invested in making honey have to build in swarm management strategies to make sure their bees stay put and that they can, in fact, make as much honey as possible.
Yeah, and I think, in a previous either question or answer when we were talking about swarming, you basically said, if there's a swarm, you lose. The beekeepers lose as a result. So do you want to tell me just real quickly the different steps that you can take management-wise to prevent swarming?
Yeah, I will. And before I do that, I want to give you just a quick disclaimer, and it's important for beekeepers to hear. Amy, as you probably know, there's beekeepers of all types on planet Earth, and some are pretty management intense, some are not. Some are, will use chemicals to control what have you, and others will not. Some will feed, some won't, etc. So there's definitely a movement at the moment called natural beekeeping, and a lot of the natural beekeepers think that bees should just be allowed to swarm. And what follows is certainly not a knock on that philosophy. If that's your beekeeping strategy, so be it. However, if you are into honey production and that is important to you and you've got to produce lots of honey for sale, especially if you're a commercial beekeeper, then you got to control swarming. And so if you think about it, Amy, swarming, because it's reproduction, is hard to, quote, control. We do our best, but occasionally we still lose. So essentially, swarm control is addressing the stimuli that lead to swarming. So I'll give you an example. If congestion is one of those stimuli that will cause a colony to swarm, you deal with that by adding more space in the hive. You alleviate the congestion. If having an older queen is a stimulus that leads colonies to swarm, then you deal with that by requeening your colonies in early spring, so you have a younger queen. If queen cells are a stimulus that lead to swarming, you might go into your colonies every 7 to 10 days and remove the queen cells during swarming season. A lot of commercial beekeepers will split their colonies a month or two in advance of swarm season because a split is essentially a controlled swarm. So you're, in many cases, it might, for lack of a better term, trick the colony into believing that it has swarms. So there's a lot of these types of things that you can do that collectively reduce those stimuli and therefore reduce the chances that your colony is going to swarm.
That's interesting. I wonder who, the person was that decided, oh, if I just go ahead and split them, Yeah,
it's funny though. I mean, it's important to commercial beekeepers. This isn't a very commonly used strategy by commercial beekeepers. A lot of hobbyists and sideliners will do kind of the more management intensive strategies, clipping queens, going in and cutting out queen cells once a week, etc. But commercial beekeepers just do a lot of requeening, they do a lot of splitting, and they do a lot of super adding to make sure there's space. And those are the strategies that they often use to address swarming.
Awesome. Do you have any other last minute suggestions for beekeepers?
Hey, if you've failed to, remove those stimuli and your colony ends up swarming while you're there, just sit back and watch it happen.
It's pretty cool.
Really an amazing process. If you're really good at it, like I am, I will sit at the entrance of a colony and catch the queen as she comes out. And
no way.
Yeah, absolutely.
I'm imagining like Mr. Miyagi. Like, do you do it with chopsticks?
That's that one. I do it with my bare fingers, and when I'm done, I put my human fingers back on.
Gosh, yep, you're bare. Jeez.
Anyway, but I'll do that, and I'll catch the queen. I'll put her in a queen cage, and then I'll sit her down, and the bees will all cluster around her, and then I've got my swarm. I've had to do this lots and lots of times when I'm in the apiary. And the reason you're able to do it is the queen is not the first to leave. She's usually somewhere in the middle. So once you see that swarm initiating, you have time to run over there and sit and wait at the hive entrance to try to catch her. But be mindful, she comes barreling out. So you have to be ready to catch her quickly.
Yeah, you must have good eyesight.
Good reflexes too.
Okay, that's fair. Well, thanks. This is probably not going to be the first or last time we talk about swarming, but we can get into it, and hopefully we can have Tom Seeley come and, talk to us about his book and about some of his other research that he's done.
That'd be great.
It's everybody's favorite game show, Stump the Chump.
Hey everyone, it's a question and answer time, and we are so thankful that you guys are still interacting with us and sending us questions. We've been receiving a ton of questions, so hopefully you all keep listening to Q&A and wait your turn, I guess. So,
I know,
Jamie,
Amy, that's kind of amazing if you think about it. People actually listen to us.
I know. They actually listen to us. I'm like, what do we talk about? So it's funny though, because all my close friends are like, you have a podcast? And I'm like, yeah, you should listen to it. And they're like, well, what do you talk about? I'm like, bees, duh.
Bees.
What else would we be talking about?
Exactly.
Butterflies, monkeys?
What else is
I don't know.
there to talk about?
That's true.
Birds.
Okay, so we have three
the birds
questions.
and the bees, Amy. You missed the joke. We'd be talking about the birds and the bees. Anyway,
well,
keep going.
today is just the bees. Maybe we'll talk about the birds next week. All right, so for our questions, the first question we have is, how soon should I put a honey super on a newly split 10 frame hive? Is there a too early, even if you don't collect the honey for a year or so?
Okay, it's never too early. So what I would say is, if I were to split a colony and see that split has, filled about 80% of the box that I moved it into, and there's clear evidence that there's going to be a continuing nectar flow, then I would put a super on, no questions asked. And so what is that evidence that I'm looking for? Well, when there's a nectar flow, bees are bringing in, nectar. And the way that you can tell, there's usually two good clues. Number one, you'll start seeing white wax being produced. So if you look down in the box that your split is inhabiting and start seeing white wax produced, that's an indication. And number two, if you take a frame out, turn it sideways, and lightly shake it, just lightly in the air, the nectar will rain out of that frame, suggesting to you that there is an active nectar flow. And at that point, I would have no problem at all supering the hive. And so I usually use those two indicators. And the longer that you're in bees and beekeeping, the more you'll learn to recognize when your nectar flows are happening and what the important plants are. So you can start using external cues to determine whether or not supering is appropriate. But in the meantime, I'd look for the production of white wax, and then that nectar raining from combs that you lightly shake as you pick them up.
So you're basically like, hey, bees, go do all this work. Let me shake out all your hard work.
That's why I say lightly, because if you give it, so basically you're turning your frames, right, so that they're horizontal. And if you shake the willies out of it, it's all going to rain out. So you turn it
the
horizontal.
willies or the nectar?
Yeah, the willies. No, the willies, the willy nectar.
Okay.
So you can also look, of course, at activity at the hive entrance. And if you're really good, Amy, and I happen to be really good, you can actually collect a bee at the nest entrance, hold it by its wings so that it cannot sting you. You can slightly squeeze its abdomen on the sides, which will cause it to regurgitate the nectar it has collected from its crop, and you can see it accumulating at its mouth parts. And if you're really, really good, as am I, you can actually taste the nectar straight from the bee's face and know where it's coming from.
Like a mama bird.
But I would, just like that. See, birds and the bees, I told you. Anyway, but I would obviously strongly recommend against doing that unless you're very comfortable handling bees.
Yeah, I was about to say, you must have been really bored. So we'll have to talk about pulling honey too soon on another question, maybe next time.
That would be good. Yep.
All right. So the next question we have, so actually there are two questions from the same person, or it's two different people named Oliver, but he had emailed us.
That's
and
also possible.
First of all, he says that he loves your jokes.
Oh,
I don't know if
really?
your wife tells those jokes and whether we call them mom jokes or dad jokes. But
yeah, no, we were just having this conversation off the air. They said the listener mentioned dad jokes. I'm like, is it only a dad joke when a male says it? I mean, what happens when a female says it? Is it still a dad joke?
Yeah, do I give good dad jokes? I don't know.
I happen to be a dad, so I guess all of my jokes are dad jokes.
All right. So he said he's finding himself treating for mites prophylactically in the spring and the fall, and he's not testing for mite levels. He's just treating. So it seems like all the mite treatments say not to treat just one hive, but everyone in the apiary. And he always has a hive above the threshold. High temperatures come quickly above that for most treatments. And because he lives in the valley, he has a pretty strong flow throughout the season with supers on. So as soon as temperatures drop in September, he typically treats. Is there something wrong with this? Should he be doing this? And what do you recommend?
Okay, so this is an interesting question. There's a lot of moving parts in it, but the main focus here is this beekeeper, Oliver, thank you for submitting your questions, is making the argument that I've got timing related treatment issues and there's a big window of time that I can't treat. So then why monitor in the first place? Why not just treat prophylactically when I can? And incidentally, Oliver, that's what a lot of commercial beekeepers do. A lot of them don't bother monitoring. They see little value in it. And their argument is, hey, look, I'm going from crop to crop to crop to crop. I'm pollinating, I'm collecting nectar or whatever. And I've only got these windows. If I were to sample during the first window, but have varroa mite populations that are low, it may be months before I get to my second window. And by that point, varroa may be a problem. So a lot of commercial beekeepers just take the prescribed treatment route. I'm going to treat at these designated time points because that's when I can, right? On the other hand, we always argue scientifically that it's best to do the other way around, to treat when as a result of monitoring and only treating when necessary. And the benefit of that is you may find yourself not having to treat as much as you think you do. And secondarily, you may find yourself having to treat more than you think you should. And I would argue the latter can happen a lot, especially if you're only treating twice a year. And so I would suggest to you, Oliver, if your current system works, then don't try to fix it. However, what I would also suggest is that there will be times in the future, potentially, that your colonies need a mid-season, a summer treatment. But since it's not in your prescribed treatment regimen, you may miss that if you're monitoring. So I think the benefit of monitoring is it tells you if you're treating too much or if you're treating too little. And believe it or not, the latter can in fact happen.
Sure. And you could actually save money by not having to purchase treatments, right?
That's correct. And Amy, I want to stress this one thing. I was at a meeting some years ago with commercial beekeepers and a few scientists talking about treating. And the commercial beekeepers who were monitoring found it necessary to treat based on monitoring somewhere between 4 and 8 times a year.
Wow.
So yeah, that was a huge eye opener to me. So basically it told me that if people are treating once or twice, they're probably under addressing varroa. But so in Oliver, in your case, if your colonies are living fine and they're being very productive and your prophylactic regimen of twice a year is working, then I would say if it's not broken, don't fix it. But I would suggest to you, if you start seeing kind of mid-season declines and things like that or lessened productivity, you might monitor around those times and see if it's because varroa populations are rebounding during a time you think perhaps otherwise treatments aren't necessary. And the last point that I'll make kind of here is, it sounds like based on your comments about temperature, et cetera, that you might be using some of the softer compounds or the organic acids, et cetera. And so a lot of people try to use those, and the treatment windows for those are usually such that it's very difficult to use them during the summer when it's so hot. So yeah, I've said a lot, and hopefully you can kind of take that in and think about what it all means. And if you have more questions about that, don't hesitate to ask us again.
Awesome. All right. So the second question that Oliver had was that he had a hive this spring with EFB, so European foulbrood. He was able to identify it because of an earlier episode that we had, which is really great. I mean,
what? Success.
I know. Exactly. That's like our biggest success story of the year.
Finally, someone has been helped by our podcast. Thank you, Oliver.
Thank you so much, Oliver. Oh, and he said some online videos, but I'm just going to pretend that all he said was from an earlier episode that we had put out.
Okay, well, we're going to claim credit for the podcast.
That's fair. Okay, so he said that he had a medium super on a hive. They went through winter. By the spring, they had laid some infected brood. He's since condensed the hive, and he's wondering if this super is safe to reuse.
So what I would argue to you, if it is in fact European foulbrood, I'm going to take your comments for granted, believe that that's the case. European foulbrood often clears up on its own when you go into a really good nectar flow. You can also treat with antibiotics, and I know that a lot of our listeners are going to be kind of split right down the middle. Probably half of you guys don't mind using antibiotics. The other half probably does mind. So what I would argue to you, Oliver, is that, my personal response is if I saw it, I'd probably treat with an antibiotic. And given that super that you're talking about is the one that carried the bees through winter, I'm guessing it's not the one that you'd be using to extract honey. And if that's the case, if you treat with an antibiotic, then reuse of that super won't be a problem at all. I mean, if there is a silver lining in the story, is that you have European foulbrood and not American foulbrood. Mm-hmm. Because European foulbrood you can treat with an antibiotic and it will go away. To get reinfected, you'll have to get reinfected. It's not just going to pop up because it's still in the hive. So if it were American foulbrood, the spores would stay around essentially forever, and you'd never be off the treatment treadmill. So in your case, I wouldn't hesitate to reuse that super if I ended up treating with antibiotics. If you don't do that, then even still reusing the super probably is not all that bad if you get through the major nectar flow, because the bees are likely to pull out of it anyway. So a major nectar flow, perhaps requeening the hive and making sure that your varroa are under control will probably solve that problem for you if you're resistant to using an antibiotic to fix it.
Awesome. Very cool. I went to so the Bee Informed Partnership had a webinar the other day about brood diseases, and something that I didn't know is that they have little take-home kits that you can use to test for EFB, which I thought was kind of cool.
That's right. So Vita, I believe, is the company that's been making them, and they work off the same technology that tells ladies if they're pregnant or not, and what you do is you collect larvae and you smush them in the little vials that they give you, and then you collect a little bit of that liquid and put it on what looks like a pregnancy test. And I forget what the answer is 1 line's negative, 2 lines positive, whatever it is. You have to read the instructions, but they make one for European foulbrood, and they also make one for American foulbrood. And both of those kits are really good. In fact, I often recommend to people to have a couple with you that you carry to your apiary so that when you suspect you have a problem, you can confirm using those diagnostic kits.
Awesome. All right. Well, thanks Oliver and whoever had asked the first question. I don't have a name on there.
Thanks,
so
guys. Really appreciate it.
Yeah. Thank you. We'd like to give an extra special thank you to the following: to our editors, Shelby Howell and Bailey Carroll, and to our audio engineer, James Weaver. Without their hard work, 2 Bees in a Podcast would not be possible. So thank you.
For more information and additional resources for today's episode, don't forget to visit the UF-IFAS Honeybee Research and Extension Laboratory's website, ufhoneybee.com. Do you have questions you want answered on air? If so, email them to honeybee@ifas.ufl.edu or message us on Twitter, Instagram, or Facebook at UF Honeybee Lab. While there, don't forget to follow us. Thank you for listening to 2 Bees in a Podcast.